We're still in chapter 24, and we're going to start off by talking about glycogenesis, glycogenolysis, and gluconeogenesis. So cells cannot store large amounts of ATP. In fact, rising intracellular levels of ATP actually inhibits glucose catabolism and promotes glycogen or fat formation. Glycogenesis is the formation of glycogen, and that's going to occur whenever there's excess glucose. And this is going to occur mostly in the liver and in the muscle cells. Glycogenolysis is a breakdown of glycogen via glycogen phosphorylase in response to low blood glucose. Liver cells, as well as some kidney and intestinal cells, also contain the enzyme glucose 6-phosphatase that removes the terminal phosphate, producing free glucose, and that glucose can then enter the bloodstream to be used by other cells. So here we have the two different, always with the cursor. Glycogenesis is a synthesis of glycogen. So we're moving this way. We have excess blood glucose. That's going to get converted to glucose 6-phosphate, then glucose 1-phosphate, and then into glycogen. The other side, we have glycogenolysis, which is the breakdown of glycogen. So we're starting with glycogen, and we have glycogen phosphorylase. And glycogen phosphorylase is going to convert glycogen to glucose 1-phosphate. Glucose 1-phosphate is then going to get converted to glucose 6-phosphate, and then glucose 6-phosphatase converts glucose 6-phosphate to blood glucose. Gluconeogenesis is the process of forming new glucose from non-carbohydrate sources, and this is going to occur in the liver. Glucose can be formed from glycerol and amino acids when blood glucose levels drop. And remember, glycerol is a component of triglycerides. Triglycerides consist of a glycerol molecule and 3 fatty acids, and amino acids are the building blocks of protein. And this is going to help protect against the damaging effects of low blood glucose levels, also known as hypoglycemia. And this is especially important for the nervous system. So as a kind of summary, we have glycolysis, which converts glucose to pyruvic acid. Glycogenesis, which is going to be the production of glycogen. Glycogenolysis, lost the cursor again. It's always about the cursor. Glycogenolysis is when glycogen is broken down into glucose, and then gluconeogenesis is forming glucose from non-carbohydrate precursors like glycerol and amino acids. Now we're going to talk about lipid metabolism. Lipids provide a greater energy yield than from glucose or protein catabolism. Fat catabolism yields 9 kilocalories per gram versus 4 kilocalories per gram of carbohydrate or protein. Most products of fat digestion are going to be transported in the lymph as chylomicrons, and they're hydrolyzed by endothelial enzymes into fatty acids and glycerol. Only triglycerides are routinely oxidized for energy, and the two building blocks of triglycerides are oxidized separately. And remember, the building blocks of triglycerides are glycerol and fatty acids. So for the glycerol breakdown, the glycerol is going to be broken into glyceraldehyde 3-phosphate, which excuse me, is an intermediate in glycolysis and eventually will enter the citric acid cycle. Fatty acids undergo something called beta oxidation in the mitochondria. And this is where the fatty acid chains are broken into these 2-carbon acetic acid fragments, and coenzymes FAD and NAD+ are reduced in the process. And the acetic acid fragments are then going to fuse with CoA to make acetyl-CoA, which is then going to enter the citric acid cycle. The reduced coenzymes enter the electron transport chain. And what that means by reduced coenzymes enter the electron transport chain, that means that the NADH and the FADH2 are going to go and drop their electrons off at the electron transport chain. So triglycerides here, which are the only lipid routinely used, broken into glycerol and fatty acids. Glycerol gets converted to glyceraldehyde 3-phosphate, which is a glycolysis intermediate. Goes through glycolysis, pyruvic acid, acetyl-CoA, and finally enters the citric acid cycle. Fatty acids, you're going to split off 2 carbon fragments, add in some coenzyme A, reduce some NAD plus and some FAD, and then we have acetyl-CoA, and that's going to enter the citric acid cycle. Lipogenesis, triglyceride synthesis that occurs when cellular ATP and glucose levels are high. Dietary glycerol and fatty acids are not needed for energy or stored as triglycerides, 50% in the adipose tissue, the other 50% is deposited in other areas. And glucose is easily converted to fat because acetyl-CoA is an intermediate in glucose catabolism and the starting point for fatty acid synthesis. Lipolysis is the breakdown of stored fats into glycerol and fatty acids, the reverse of lipogenesis. Fatty acids are actually preferred by the liver, cardiac muscle, resting skeletal muscle for fuel. And remember that the brain will continue to use glucose. So the brain wants glucose, whereas the liver, cardiac muscle, and resting skeletal muscle actually prefers fatty acids. Lipolysis is accelerated when carbohydrate intake is inadequate. So we talked about beta oxidation on one of the previous slides, and when beta oxidation is occurring, it's going to result in the production of large amounts of acetyl-CoA. Now, that acetyl-CoA can enter the citric acid cycle, but only if there is enough intermediates and that are available. If the intermediates are not available, then acetyl-CoA can accumulate. Accumulated acetyl-CoA can be converted by ketogenesis in the liver to ketone bodies. And so this looks a little complicated, but it's not really that bad. There we go. So triglycerides split into glycerol and fatty acids. Glycerol gets converted to, and I should say this is lipolysis, and this gets converted to glyceraldehyde 3-phosphate, which then pyruvic acid, acetyl-CoA, citric acid cycle. Also, lipolysis, fatty acids, beta oxidation produces acetyl-CoA, which can go into the citric acid cycle. If we have an accumulation of acetyl-CoA, we can have ketogenesis occur, and we can form ketone bodies. Now, you notice that these arrows are going both directions, and that means that this does go both directions in these pathways. And then again, from the citric acid cycle, we go to electron transport chain where we make the ATP. All right, quick summary of lipid reactions. We have beta oxidation is going to convert fatty acids into acetyl-CoA. Lipolysis breaks down lipids to fatty acids and glycerol, and lipogenesis forms lipids from acetyl-CoA and glyceraldehyde 3-phosphate. Lipids play many roles in structural materials. We've actually already talked about this. Phospholipids are important for cell membranes. That goes back to AMP1, the phospholipid bilayer, as well as myelin sheaths. Cholesterol is used for cell membranes to help stabilize it against temperature changes and steroid hormone synthesis. In addition, the liver synthesizes transport lipoproteins for cholesterol and fats and synthesizes cholesterol from acetyl-CoA. and finally uses cholesterol to form bile salts. Now we talk about protein metabolism. Proteins do deteriorate, and they need to be continually broken down and replaced. Amino acids are recycled into new proteins or different compounds. Proteins are not stored in the body. When dietary proteins are in excess, amino acids are either oxidized for energy or converted to fat for storage, but they are not stored as proteins. So we have the degradation of amino acids, and the goal is going to be to produce molecules that can be used for energy in the citric acid cycle or converted to glucose. The first step in amino acid breakdown is deamination, where the amine group is removed and then converted to pyruvic acid or one of the keto acid intermediates of the citric acid cycle. There are 3 events of amino acid degradation. They're transamination, oxidative deamination, keto acid modification. I'm not going to ask any questions about this on the exam. About this slide on the exam. Now we have protein synthesis. Amino acids are the most important anabolic nutrients. They form all the proteins as well as the bulk of functional molecules. Protein synthesis that occurs on ribosomes is hormonally controlled. For example, we have growth hormone, thyroid hormone, sex hormones. And then we talked about this in the very first video. Synthesis is going to require a complete set of amino acids, and essential amino acids must be acquired in the diet. During our lifetime, depending upon our body size, we can synthesize anywhere between 225 and 450 kilograms of protein, which comes out to somewhere about 500 to 1,000 pounds of protein. These are the questions for this segment. This is all I have for you for this video. Have a good day.